Thank you for the introduction and for the chance to talk a little bit about our imaging work. A lot of this data that I'm going to present this is the first time I'm presenting any of it. So I welcome any feedback, thoughts. This is sort of a first foray in thinking through a couple of new ideas. So this first, I want to preface this with, I'm a clinician to start with and a researcher afterwards. And I'm trying to think a lot about not only the science, but tools that we can create that are clinically useful to help our medical decision making around the boys with, and men and women with ALD. So this first analysis we did in diffusion tensor imaging, which I know Dr. Pierpont talked about a little bit earlier today. What we wanted to do was create, like I mentioned, a tool and a way to analyze ALD lesions and the normal appearing white matter that are around ALD lesions to hopefully tell us something about whether or not a patient's going to have progressive disease versus is their disease stable, and perhaps can we actually predict disease. So if you can imagine, this is this brain MRI in midsection, and you can see in the splenium of the corpus callosum this tiny T2 hyperintensity right here. This is a early stage blooming cerebral ALD lesion. And as mentioned, the corpus callosum is essentially a bridge, right? It's a bridge of fibers from the left and the right side of your brain, and they swoop back and forth and connect both of your hemispheres. Early cerebral lesions in ALD usually start right in the midline, right in the back of that bridge. So if you can imagine us taking slices of that bridge and essentially measuring using diffusion tensor imaging different metrics over those spatial time points, you can essentially map out the change in any value that you want. So DTI classically puts out 6 major values. They're all, for people who are into linear algebra, they're all eigenvectors that are orthogonal to one another, and then you can generate any metric you want. But fractional anisotropy, the measurement of order in the brain, is probably the most famous one and the one that's most used clinically. So we can measure the FA values along the corpus callosum in the normal appearing white matter from within the lesion, then close to the lesion, then getting farther and farther away. And so we did this in cases of patients who had active inflammatory cerebral ALD and patients who had spontaneous disease arrest or self-stabilization. And so what we found is on top, if you look, that's the FA values across space, proximal to the lesion to distal or close to far. And we noticed that in patients who had lesions that remained stable, their FA, their fractional anisotropy, close to the lesion was high. And that tends to happen if you have a cerebral scar. There's not a lot of movement of water. There's a high degree of scarring and organization. It tends to drive your FA value up. And then as you go distal, right, it sort of normalizes because you're in normal appearing white matter. So you get this negative slope. Conversely, if you are a patient who has active demyelination, the FA value in and around the lesion is actually lower because there's edema in there, there's swelling, water's moving in more directions than it usually would if it was just restricted to normal myelin. So we're sort of positing that, and we see this relationship amongst pretty much all of the subjects that we tested. So, thinking about clinical tools, are there things that we can do in addition to just the classic MRI sequences that we're doing that can tell us something about how at risk a patient is and whether or not we should be thinking about gene therapy or transplant or enrolling them in a clinical study for that matter? So we repeated the process and said, well, okay, what if we exclude the lesion? So we start, like I said, a little bit in front of the lesion. We're not including the T2 white matter hyperintensity. Does that same relationship still exist? And it does. So we're starting to generate data where we can measure parts of the brain that we don't think are diseased, but we can measure changes in between groups of progressors versus non-progressors. So we're working, we started this work a couple years ago. This is while I was still at Cornell and sort of moved it over to Kennedy Krieger. But this work was submitted and accepted at ISMRM, so I'm gonna give a talk about that to the Brain Imaging Society, and this is one of the biggest, if not the biggest, brain imaging society, and it's an opportunity to talk to them about ALD, and it happens to be in Hawaii. Yes, I'll dive on that grenade for us. What?
I'm paying for this.
No, I'm paying for this. I don't think you pay for anything from me. Okay, so that's diffusion tensor imaging, a pretty easy tool. We can program that. We can stick it into a pipeline, and you get a slope. You get a number, and it helps you perhaps discriminate what's happening in some of the patients. This is a different imaging technique. It's called radiomic texture analysis. And actually, the person who did this analysis is sitting right there in the back of the room. That's Shahnaj. So she is one of the graduate students at Cornell who has been working with me for about the past year on some of this data. And what a texture analysis does is it looks at, you can look at gray matter, white matter. We're interested in white matter, of course, at least at first glance. And in the same way, if you do a biopsy of a tumor, for example, and you put it on a slide and you look at the slide, you can look for features of those cells that make you think, well, this is a benign tumor versus this is an invasive tumor. This is or something somewhere in the middle. And so I want to liken it to that. We can look at the texture of the actual MRI scans. Using a little bit of coding and more linear algebra, to get a sense of is there something that's going on in the brain that we just can't see by just looking at the MRI. So this is subvisual. You can't see it, but we can measure it. And it sounds like magic, right? So this is called a radiomic texture analysis. The GLCMs, what you essentially do is you take all of the gray matter pixels. You take all the pixelations in the MRI, and you essentially drop a matrix onto it, and then you look at the variability in the pixel intensity between neighboring pixels, and then you can essentially run a couple of different types of equations that give you a sense of what local variation looks like, how uniform perhaps the signal actually is, how homogeneous it is. But what we're interested in is this term a physicist must have really put this together when they did the MR physics of it. The measure we're interested in is entropy. So it's a measure of disorder, but you can't see it on the MRI. You're just looking at gray and white matter. So we're interested in this measure of disorder. And so what Shahnaj did was she went and looked at, we're used to looking at T2s, but you can do this with basic T1 imaging as well. So she looked at cerebral ALD lesions. She looked at the normal appearing white matter just outside the cerebral ALD lesions, and then she looked at distal normal appearing white matter higher up in the brain where there's no sort of local disease. And you can essentially just start to take those measurements. And when we took those measurements, a couple of things came to the fore. So the measurements within the T2 lesion differ between patients who have progressive disease versus non-progressive disease. So if you're an imaging person and you're reviewing MRIs and you're looking at a T2 cerebral ALD lesion, more or less they look pretty much the same. And one of the ways that we try to tell whether a lesion is active is we give contrast, and if it contrast enhances, it has an 80% probability that it's going to progress. And then there's also the relationship with age. But this may be a tool that we can test out where just getting basic T1 and T2 MRIs, we can do this analysis, and we can tell you if somebody walks into the clinic and has an abnormal brain MRI, maybe they don't have any prior imaging, we can tell you if it's old or new, or if it's more predicted to stay stable for a while versus we think it's actually going to progress. So things, more clues to help guide our management. And then that same relationship was found just outside the lesion, and then it was found elsewhere in the brain. So it's starting to tell us, and I think, Dr. Pierpont, you probably have this, this was mentioned earlier, there's clear symptomatology that's related to the lesion itself. Then there's other stuff. enough, right? There's symptomatology and signs of disease that are not accounted for by this thing that we focus so much on. So your work and then some of the work we're doing now is we're trying to look, hence the subtitle of the talk, beyond the lesion. So Shahnaj did a really good job with this, and she's working on writing it up. Hopefully we'll have that published soon. And it was accepted into the meeting for the Organization of Human Brain Mapping. So that's been accepted, and then I sent it to ANA. So hopefully they looked favorably on it. Okay, so it's like a marathon. So this is the last technique I'm going to talk about, but this one's probably going to take the most time. And please stop me, please interrupt me. But there's this type of imaging called quantitative susceptibility imaging, or mapping. And what I'm going to talk a little bit about is what that actually means and how it applies to ALD. So I'm going to read this, and then I'm going to explain it. Quantitative MRI technique that decodes the distribution of tissue-specific perturbations in the local tissue magnetic field induced by an externally applied magnetic field. So if anybody took physics or organic chemistry in the past, there's going to be a lot of PTSD in this talk. And I gain a little bit of pleasure by torturing everybody because I had to go through it. So. In the most basic sense, all the molecules that are in your head are, to some degree, susceptible to magnetic fields, right? We're made of atoms and molecules and organic things, and every atom, molecule, compound that you have in your body generates a small magnetic field to some degree, whether it's a tiny one or a larger one. So if everyone remembers, like from physics, like the right-hand rule, like things spin and then they generate a current, they generate a magnetic field. Every molecule in your brain does this. If you were to measure every molecule in your brain and apply an external magnetic field, like an MRI scanner, some of those molecules may be susceptible to the outside magnetic field, and some may not be that susceptible. So that's where the word susceptibility comes in. So, for example, myelin is hydrophobic. It means it doesn't like water. It folds in on itself. It has no paired electrons, so it has no net magnetic field of its own. So this is referred to as a diamagnetic structure. If you try to measure how much myelin moves in response to an external magnetic field, like an MRI scanner, it doesn't move very much. It's negatively susceptible. In contrast to that, something like iron, which exists for people who remember organic chemistry and general chemistry, exists in an oxidized state, or it's bound to transferrin in our body. And because it's not fully valent, iron, all atoms, they spin, they generate their own magnetic field, and then when you apply a magnetic field, something like iron is highly susceptible to an external magnetic field. Okay? So that's known as a paramagnetic molecule. So everyone's with me so far, right? For the most part? Okay. So this is brain, external magnetic field, and I sort of just talked through this, but just to summarize. So there's negative susceptibility, and we think that measures myelin. There's positive susceptibility. We think that measures iron. If you look in other neurodegenerative disorders, they've sort of proven this: MS, Parkinson's, Alzheimer's. And so if you talk to the real imaging people, not the ones playing one on TV, they will tell you that it's myelin and iron. So QSM is the map of how susceptible all these little molecules are to the magnetic field. And you can separate it out into its component parts. So this is what we call source separation. You can separate it out between the molecules that are negatively susceptible, myelin, and the ones that are positively susceptible, what we think is iron. And for people in the imaging world, get a sense, like, this images out myelin pretty well. Like, it's a fairly high contrast image. So we have our two source maps that we think is myelin, we think is iron. We haven't proven it in ALD yet. And so what we did is we looked at 100 cases of normal brain MRIs from boys and girls, and created an atlas. We smushed them all together, gave them an average. So it's essentially your control group. And then you can take your ALD MRI and put it on top of that atlas and look at where there are changes from the control group. Good. And so this is what we see. I'm going to orient you really quickly. So the top row, this is one patient, one time point. This is a different patient, different time, all at one time point. This patient has a lesional inflammation, right? So active demyelination. This patient has no active inflammation. This patient's MRI has remained stable like this for the past 3 years. So somebody with progressive disease, somebody with non-progressive disease. This column is the QSM negative map. This is the Myelin map. QSM positive, so this is the iron. But I just want to focus and draw the point that if the brains match the controls, everything would be sort of this green-yellow color in the middle. And I changed the coloration for the next slide just to make it easier. But I'm going to walk you through what I think we're seeing. So here's the overlay. So this is the patient with active cerebral disease. Here is their iron map. Red is 2 standard deviations higher iron content, what you think is iron content, then controls. And blue is 2 standard deviations or lower of iron than controls. And then if you overlay it, you can see where the white is. There's lesion, there's normal carrying white matter in this little pocket right here, where there's low iron content. And then just outside the lesion, where in the normal carrying white matter, the iron signal's high. And you always want to take an opportunity to try to one up your mentor. So, we've seen this before, right? So my hypothesis here is, and this is Dr. Eichler's work that he alluded to earlier, thinking about microglia and what they're doing in and around a lesion in the cerebral ALD brain. And he points out that this region number 4 here is where it's thought that microglia are dying, and there's this band around every lesion where there's no microglia. And then in the center is our blood-borne macrophages. So if I put these side by side, the spatial distribution of the signal, I think, matches what we know about the pathology. And it appears to be that the iron signal matches the spatial distribution of microglia in cerebral ALD. So Dr. Eichler put this together in a paper, and he broke up the cerebral ALD lesion into multiple zones. So this is the center of a lesion, complete demyelination, active demyelination. This is where we see the contrast enhancement. Perilesional, so microglia are thought to be undergoing apoptosis, they're dying, so there's no microglia in the perilesional space. And then extralesional is activated microglia. So if I map on what I see in the QSM signal, everything from the perilesional space downwards, it's blue, right? The QSM is negative. The QSM positive is low. But the QSM positive signal is increased in the extralesional space. So that's one half the story, and I'm going to put this all together. I promise. I will get there. Thank you for taking this ride with me. So now let's focus on the myelin signal. So this one over here, you can see it outlines the cerebral ALD lesion pretty well, right? There's a drop off, there's demyelination, so the myelin signal is gone when it becomes blue. But if I also want to mention one more thing. As you can tell, I'm still working through learning a lot about this and what I think these images mean. The elevations and drop-offs in the myelin signal tend to be around the lesion itself, or where we think the lesion's going to form. The iron signal is seemingly kind of random, right? It's just in the normal inherent white matter outside the lesion in no particular distribution. So going back to the myelin signal, the QSM negative map. If you look all the way on the left, you'll see right here there's a tiny budding T2 lesion. if we look at the processed QSM, where the lesion is, it's cold, right? There's a drop off in the myelin signal because that area is being demyelinated. But in the perilesional normal appearing white matter, the signal's super high. It looks hot. So if you look here in the normal appearing white matter, there's no lesion here, but the QSM is telling us that something is happening to the myelin that's increasing its susceptibility to the magnet. So this is an increased susceptibility signal. And you can see the scale over here. It's like once you get to 2 standard deviations up, it's about red. Dark red's 3.6. So iron, myelin, and so as it turns out, our brains process lots of iron. Myelin takes in lots of iron. Microglia take in lots of iron, metabolize it. Activated microglia suck up a lot of iron to do various things like help kill off bacteria and things like that, and pathogens. So what sort of brings us? Oh, and the last thing is, as we age, our brains accumulate iron that we can see over time. So our caudate nucleus, our globus pallidus, the red nucleus. If you look at a kid and follow their MRIs from childhood into adulthood, you'll see normal iron deposition in the brain. It happens to all of us. So there's as it turns out, there's a lot of iron in our brain. So how do you tie this all together? Well, if I'm thinking about the apoptosis story and I'm relating it to iron, there's new work looking at this independent mechanism for cell death called ferroptosis. So in different disease states, iron comes into the brain, or whatever cell type you're thinking about. We think about neurodegenerative disorders. And what ends up happening is, for whatever reason, and I can tell you why I think it's happening in ALD, but for whatever reason, we can't metabolize the iron well enough, and it starts to build up. And what ends up happening is reactive oxidative species, so unpaired electrons that are stuck to oxygen or peroxide, start to build up. And so the downstream effector of ferroptosis is what's called lipid peroxidation. So the lipid bilayer that forms every membrane of every cell in our body starts to have peroxide moieties essentially get stuck to it, and that causes the cell membrane to start to break apart. And when the cell membrane breaks apart, all the contents get released and the cell dies. So this, I think, might be the unifying mechanism. So that elevated myelin signal, normally if we have abnormal reactive oxidative species in our brain, which we get all the time because we can breathe oxygen, we have mechanisms for reducing that oxygen back to water. In certain states, that oxidative balance of being able to reduce it back to water or put oxygen somewhere where it won't do damage, that balance is off. And I think in ALD there may be a role for this. These reactive oxidation fragments, when they sit on the membrane of myelin, it increases the myelin susceptibility because they have unpaired electrons, and unpaired electrons really like being moved around by a magnet. So here's what I think is going on. This is the hypothesis. We have not proven it. But taking all the pieces of the story together, all the action is kind of right here. And we also know that the spatial arrangement of a cerebral ALD lesion is also the temporal sequence of events. So what I'm going to posit is essentially, is microglial ferroptosis an early pathogenic change in cerebral X-linked adrenoleukodystrophy. So I sort of went through all of this, figuring out the imaging, and I thought, oh my gosh, I think I figured something out. What do we know about reactive oxidative species, oxidative stress, and lipid peroxidation in ALD? I didn't think we knew that much, but of course, the people who are in this room did this biochemistry 10, 15 years ago, and were way ahead of the game. But this one paper in particular, Adrenoleukodystrophy, Oxidative Stress of Mice and Men by James Powers, talks a lot about oxidative imbalance in patients with cerebral disease. And one of the salient conclusions was they found evidence of lipid peroxidation in normal appearing white matter beyond the lesion. Lipid peroxidation is also a self-replicating process. So it will spread until it uses up all the myelin in the brain, which sounds kind of like the disease. So my whole sort of takeaway from this is, I think if it is in fact iron, maybe there's evidence of, just like in other neurodegenerative disorders, a disordered myelin metabolism in the brain, and that can lead to one of this sort of pathogenic mechanism that may be contributing to the onset and spread of cerebral ALD. So we submitted this also to the ISMRM. We're going to have an opportunity to talk to the radiology community about this and present this work. And then we're going to turn this ferroptosis story and we're going to bring it back to the lab, do some initial experiments, and hopefully take this idea and try to move it to the next level. Okay, and I have lots and lots of people to thank, but I will start to take questions now.
Beautiful work, Eric. I mean, it sounds like you're answering the question that I only got a question in the title with years ago. So much kudos to you. I guess some of the things that are going through my head are, is there a way of, doing post-mortem tissue MRI similar to what Mario did years ago that might pick up, some of these iron associations with apoptosis. The second is there is a necrotic core in ALD, in cerebral ALD, and maybe it's in at least in the images you showed, which was relatively early stage compared to some of the autopsy cases where a true necrotic core is visible, but shouldn't that also show iron deposition? Because I didn't see that in the images.
So lots of questions. I think you're exactly right. Dr. Van Haren, I showed him this data yesterday, and his first question, which is the right one, is, well, where does it go, right? So if you have a necrotic core, all this microglia, which are now dead, have emptied out their insides. Perhaps there should be iron deposition in the necrotic core. We don't have the answer. So one of the things we're going to look to do is do postmortem imaging, do some histology, and see if we can match the spatial distribution of what I think we're seeing on the scan with what's actually seen under the microscope and on postmortem MR imaging. It may be something else. What led me to this particular hypothesis is just all the pieces sort of lined up, but it may turn out to be a totally different story, and we're completely open to that. Yeah.
Thank you for that great talk. I'm not going to ask about the QSM because I'm going to be processing that for a while, but it's really, really cool to hear about. My questions are really surrounding the idea of clinical applications. Mm-hmm. And, I'm thinking about what you presented with DTI, and you've really helped me think about things in a different way, thinking about kind of moving from that lesion core more distally and how that kind of gradient might be actually really illuminating and really interesting to be considering. I think, in terms of clinical applications, certainly being able to distinguish, help distinguish in a situation where we see a lesion and we're not sure if it's active or not, we don't see gadolinium, could something like this be helpful? So I guess that's my first question is, do you see possible clinical uses in that example? And then kind of beyond that, thinking about post-treatment, whether any of these techniques could be useful in thinking about how we really want to stop this disease in its tracks, right? So if we see changes more rapidly in a good direction, in a way that we're like, okay, we don't think this is active anymore, that could be a really good mechanism of tracking, response to therapy.
Absolutely. Yeah, I think and there was the case that was brought up yesterday about a mother whose son's disease was progressing and getting bigger without showing gadolinium, right? And so the question is, well, what would these metrics show in that case? Would they have been consistent with a progressor despite the fact that there's no gadolinium enhancement? And then in doing the texture analysis, looking at the texture of the lesion itself on T1, T2, whatever you want, and does it fit the profile of somebody who's a progressor despite not having the gadolinium? I certainly think that would be helpful. And then the flip side, of course, right after transplant, we all sit there for 6 to 9 months going, all right, this MRI, I hope the gadolinium's gone, and sometimes it's still faintly there, but gadolinium in post-treatment means something different, right? You've treated the lesion, and so that gadolinium that's there, it doesn't mean the same thing, obviously, pre-treatment. But if we added these metrics in, your slope looks like a non-progressor. The texture of your lesion from before treatment to after has changed. Then you get more certainty in thinking, okay, I think your disease is going to stop, versus did you escape transplant and we have to do it a second time, or in the era of gene therapy, for whatever reason that the gene therapy doesn't take, then you have to go to transplant, or vice versa, right? However that works. I see applications in both of these things, so I try to keep my research centered around things that are usable and are done on acquisitions, DTI aside, but are done on standard acquisitions, T1, T2. Right? Everyone does them, so theoretically we can apply these tools to every scan that comes in the door, certainly.
Along those lines, I just want to point out, this is all without gadolinium.
What?
This is all no gadolinium.
Yes, exactly.
Included.
Right.
Just for everyone who worries about giving too much gadolinium to children, I just want to point that out.
Thank you, and thank you for bringing that up. That's one of the things that we're trying to think about is how can we image these kids without giving them gadolinium repeatedly, and do we have different sequences that can give us different types of information? And so part of the study, right, is going to be, when we put the models together, is how much does it correlate with gadolinium, with the idea that theoretically someday in the future maybe we don't need it because we can do all these other metrics, it gets spit out on a test and you go, you're a progressor, non-progressor. And I didn't need gadolinium to tell me that. We can do gadolinium in the MRI right prior to transplant to make sure the blood-brain barrier is in fact broken down, or do Patty's perfusion imaging. But this would be a nice way to get around repeated gadolinium exposures.
So I understood about 10% of that. The question I'm going to ask does pertain to the 10% though. So this study, you have to do the overlap of a normal brain to measure what you want to measure, right?
Yeah.
Okay. So is that what's called the mapping? Am I using the right term?
Yes.
Okay.
Mapping means exactly what you think it means.
So my question is then, when you're doing the comparisons, does the age of the brain have to matter? Because I know the younger you are, you're less likely to find normal brain scans, right?
You understand much more than 10%, I promise.
Okay.
The reason we pick 5 to 10-year-olds was because in normal brain development, as you alluded to early in life, brain MRIs change relatively rapidly in the first 2 to 2.5 years. So a baby brain is very watery. Once you get to 2.5, you're like 90% of the way there in terms of myelin development, and the appearance on the scan is much different. Once you hit like 5 to 10, that growth curve, that change, has kind of flattened out a little bit. The terminal zones of myelination are still kind of coming in, but for the most part, your brain's mostly myelinated. So this was a first pass of taking 5 to 10-year-old normal cases as far as we can tell, and sort of lumped them together, squished them together, flattened them, and came up with a template. What we want to do next is actually break it down by age. So, for example, we'll have 30 cases of a 5-year-old, 30 cases of a 6-year-old, and then generate these templates so that when you hit that age, you get a perfect age match.
Okay.
Because as I mentioned, our brains are dynamic, right? They're they myelinate quickly early in life. We undergo pruning, so we lose axons. We accumulate iron over the course of our lifetime normally. So there's a lot of things that are happening that we have to account for. So one of the things that makes ALD and tracking ALD or any progressive neurodegenerative disorder difficult over time, especially in childhood, is you have to control for all the developmental trajectories of all these normal biological processes before you can say, oh, my disease is doing this or this. You know, you want to make sure it's not an artifact of just normal development or you're misinterpreting something.
Okay. And
So-
the normal iron that you accumulate as you age, does that have anything to do with dietary iron? It's totally, we're talking about different things.
Everything.
Okay.
I think your diet has in some sense, everything you put into your body, right? Your body has to figure out how to process.
Okay.
But there is a normal range and trajectory of iron deposition in the brain that I don't know if somebody ate liver forever and would they have higher iron content in their brain? Perhaps I don't know that's been measured, but yeah, it just seems to be part of normal aging. Okay.